Our next lecture, we're going to talk about the ABO system. Oh, by the way, ABCs. Everybody, know why I picked that song today? AB, we have no C blood group. Actually, we do have a C blood group. We won't get to that until next week. Wait, ABO? ABO is the only blood group system where antibodies are predictable, consistent, and naturally occurring. It's the only blood group that we're going to find that have those three things about them when the antigen is lacking. For example, I'm type A. I know if I'm type A, I have anti-B antibody. I know that if you tell me you're B, you have anti-A antibody. I know if you tell me you're O, you have anti-A and anti-B antibody. So the ABO group is the only ones that we know of, the only group that is predictable, consistent, and naturally occurring. It's one of the most important blood groups. In vivo, in the body, it can cause fatal reactions. I've told you a couple of the articles we're going to talk about when we get to transfusion reactions and stuff like that. There's a couple of articles where somebody gave the wrong type and people have died. It happens. It's gotten better over the years because we have all these things in place. Remember in lab last week I talked about we always put the tubes in the same order in the rack. We always put our reagents in the same order. We always do 1 tube per rack. We have all these rules in blood bank to keep that from happening. That is why. You guys learned in phlebotomy. When you draw a blood bank tube, they get a special band. Now, not every place does that still. Some places it's still built into the barcode, but a lot of places still give that special blood bank band that number follows this number that follows through. Remember you put that number on the tube? We'll get some of these when we get to some of these cross matches where you'll get that string of numbers back and you're going to follow that number from that patient on all the paperwork and everything all the way through. ABO compatibility between donor and recipient is critical. This is one that can cause a lot of problems if we don't. Hemolytic transfusion reaction due to a major blood group incompatibility is one of the JACO's defined sentinel events. You guys remember talking about JACO in BLS? Joint, oh boy. I don't remember what it stands for. It's a health organization. It's the Joint Commission Association of Health Organizations, maybe? Something like that. Anyway, they're the ones that oversee the hospital. The sentinel events are ones that are like instant report, stop, and investigate. Giving the wrong blood is one of them. Losing a baby is one of them. Cutting off the wrong body part is one of them. Like during surgery, if they cut the right leg, it was supposed to be the left. That's one of them. So it's all these events, and if something like this happens, it's some of these major safety things. So these sentinel events are these major safety things put in. Giving the wrong, getting a hemolytic transfusion reaction due to a major blood group incompatibility is one of the events. So when do we do ABO grouping? Transfusion recipients. You're going to get blood. I need to know what type you are to make sure I give you the most compatible. Blood donors. I need to know what they are to put on my shelf so I can give it to the right donors or the right recipients. Transplant candidate and donors, we blood type them because there's a lot of ABO antigens on all the other organs sometimes. So certain organs, it's really important to make sure we have a nice good match. Certain, it doesn't matter as much. Pre-transfusion workup. Anybody that I'm giving a transfusion to is going to get an ABO. Prenatal patients, one of the first things they do, when you go into the doctor once you're pregnant, the first appointments, they draw your blood. They want to find what your blood type is. Because that's going to help them keep an eye on you throughout the pregnancy to try to help keep baby safe. Newborns, sometimes, yes.
So for the prenatal patients where they're drawing the mother, does that blood actually go to a blood bank, or do they usually test that
We test that on site. So if you are at the clinic and I draw a new mom, that blood just gets tested on site, unless there's something weird, and then it goes to a reference lab if we're worried about something. Most of the time it's just tested on site.
Okay.
Newborns sometimes, if we know that there's been problems along the pregnancy, science will test to make sure that those problems are stopped. My daughter, we had problems all throughout the pregnancy, not related to this, thank goodness, but other problems. The minute she was born, she thrived. So, you know, a lot of times if we know we have problems along the way, we're going to test to make sure it's going to stop once they're born and they'll thrive. Make sure it doesn't continue on. And paternity testing. Now, we all know there's better options out there for paternity testing, but it's a quick and easy one still, so we really don't use this for paternity testing. But we do use this when we do sometimes test dad if we're looking for how sick mom is going to make the baby. Because we want to, you know, there's things that we want to know if dad's homozygous, heterozygous, what's going on. We'll talk about that with the AHDN. Sometimes that becomes problematic because who's the daddy, right? Sometimes they don't tell us it was a sperm donor. We're doing all these workups and things just aren't matching. We don't know what's going on. And we find out they had a sperm donor or an egg donor, you know. So there's things that happen along the way. So, Carl Landsteiner, that name we need to know, right? Landsteiner. 95% of blood bank, that's going to be the answer when it's a who. He developed this in 1900. We talked about this last week. Received the Nobel Prize for Medicine that year for developing this. Mixed the serum in the cells of researchers in his lab and found 3 patterns of agglutination. The 4th pattern was found about a year later. So, what they did, these are the different doctors in the lab. I'm going to try to pronounce them, but I'm going to tell you my German isn't very good. And what they did was they took the plasma of Dr. Stork and put it with the red cells of Dr. Stork. Should be fine, right? Your own blood, your own plasma, you should be able to put it together without a problem. So it was fine. But when Dr. Stork's plasma with Dr. Pleshnik's cells, it agglutinated. Dr. Shirley's cells agglutinated. Dr. Erdheim's cells agglutinated. Dr. Zarich's cells agglutinated, but Dr. Landsteiner's did not agglutinate. That was interesting. But what if we took Pleshnik's plasma and mixed it with all those cells? And Shirley, Erdheim, so we mix all their plasmas with the cells. So if you follow, this is their own cells and plasma is this line through here, so we expect not to see any agglutination that way. That's kind of interesting. Why is it agglutinating with some and not with others, right? This is where we found some of this out. So his rules came out to be, a person does not have antibody to his or her, this is back in 1900 though, so it was only his, right? And his own antigens. Okay? You do not have antibody to your own antigens. Makes sense. With regards to ABO, folks in there, each person has the antibody to the ABO antigen he lacks. So, I have these to hand out. Will you do me a favor to pass these around the room? Good one. So, group A here, you can see has pink circles on the outside. I'm going to do this way. Seems to have, my pieces have fallen apart here. You can tell I don't play with Legos much. Wait, now I'm going to build this. How do I do this? This way? I didn't have pink circles, but I had pink squares. So you can tell the antigens for A look one way. Group B, these little green / blue antigens on the outside of the wall. Now remember, there's over 300 blood group antigens. So I'm being super simplistic when I'm drawing these pictures to show the antigens on the outside. Group AB has A and B antigens on the outside. Then we have group O. If we look down here at group O, see how they have these little spikes but no antigens? Lack of. That's what I'm talking about with having the amorph. They have a lack of the sugar that creates that antigen. Antibodies in plasma. I know if I have a group A, I'm going to have B antibody. I made it yellow because in the lab, yellow is B, anti-B, and blue is anti-A. So what we're looking for is if I try to go to put this on, it's not going to quite fit. Again, it won't fit. It won't go against the A. But if I suddenly get B blood, it's going to fit. Well, it's supposed to fit. It's going to fit. See how it kind of sticks in there like that? Anti-A, I'm going to, whoa, what happened?
I think you're recording, so.
Yeah.
Okay, let's go back. Oh. Okay, let's not stop. The other one must have finished rendering, yes.
I didn't get one of the sheets.
Oh, were the other extras? Oh, what was it? There should be plenty. I think I have like 5 extras. Did everybody else get one? Okay. Thank you. So, if I am A, I know I have group anti-B. If I am B, I know I have group anti-A. If I am A and B, I can't have anti-A or anti-B, or my antibodies are going to be destroying my own cells. And if I'm O, because I don't have any antigens on my cells, I'm going to have both anti-A and anti-B. So we're talking about ABO, that's what we're talking about. Blood group O antigen, and I put it with anti-A or anti-B. O antigen, antibody in serum, A and B, and anti-AB. O is kind of special because O has an anti-A comma B IgG component often, and it's a little bit different. We're just going to kind of like ignore that piece right now. That's why it's in parentheses. The main ones you have to know is the A and the B. So if I start putting on, if I put this serum, it's not going to glutenate with A or O, but it's going to glutenate with A, B, and AB. My A here, I have anti-B antibody in the serum, so if I take that serum, I put it with O cells, there's no B antigen on the O cells, so it's not going to agglutinate. There's no B antigen on A cells, so it's not going to agglutinate, but there is B antigen on B and AB, so it's going to agglutinate. Anti-A cells with A, it's not going to, with O, I'm sorry, it's not going to agglutinate, but if I put anti-A in cells with A antigen, it's going to agglutinate. B, it's not going to, and AB is going to. AB blood type has no A or B antigens. I'm sorry, it has A and B. Repeat. AB blood type has A and B antigens. It has no A or B antibodies. So I can put that plasma with anybody and it's not going to agglutinate. Some of the incidence of population, this is a Caucasian population. In a Caucasian population, O is going to be your highest value, highest, most common. A comes in second at 40, B at 10, and AB at 5%. Only 5% of the population is AB. And part of that, if you think about it, you know, B's are only at 10, so just think probability, right? So you have to have the right parents to make that one. And so with this being so high, that ends up being so low. Now you can tell there's some differences. Mexican Indians, about 90% are O's. So depending on where you are in the world, you're going to want to, know your donor population and your recipient population. Make sure you have enough on the shelf. If I was, in an area where 90% of my people had O blood, I want to make sure most of my blood is O on the shelf. I don't need a lot of A floating around. Versus in Wisconsin, where these are pretty close, I need to have some A. Now, this is just for information. It's just kind of a fun map. Percent of populations that has the A allele The white is very, very low, the dark blue is very, very high. So you can see there's a lot of A here, and then the pinks are kind of in the middle, a lot of A down there. Bs, same thing, like not many Bs, but then look over here, look at some of these dark red spots, 20 to 25% over here in the Bs. And Os, you know, here we have a lot, some here, some there, but then look at all those areas that they don't have many Os floating around. Just kind of interesting. Migration of people throughout history and ancestry has always fascinated me. That's where a lot of this comes from. ABO typing reagents. Forward reagents, we have antibodies, anti-A, anti-B, and anti-AB. See the colors? They're all color-coded. That's one of those little things that we do to keep people safe, keep things safe. We're going to use these reagents to test for antigens on the red cells. We use antibodies, known antibodies against unknown antigens. These can be done in a slide or in a test tube with a patient 3% suspension. We do everything in a test tube with the exception of donor segments. In lab this week, we're going to type patients and donors, so make sure you read all the way, I think it's page 16 through 22 in your lab manual. Donors get done on slides, but patients we always do in tube. So this is the forward typing. Forward typing is where we're typing for the patient's antigens. So here's some very basic slides of what it would look like. The blood sample with anti-A, anti-B, and anti-D. This is agglutination. This is not agglutination. This is what it looks like when you do it on a slide. So this patient, it agglutinates anti-A and anti-D, so this is an A positive. This patient agglutinates anti-B and D, so this is a B positive. This one agglutinates anti-A, anti-B, and anti-D, so that's an AB positive. This one doesn't agglutinate anything, so this is an O negative. Do not memorize. Do not focus on the books. This is when you're learning these agglutinations, you have to understand, think through what's happening in the tube. Don't just memorize what's going on. Now, reverse typing. Oh, yeah.
Why do you do the slides versus the tube for the donors?
Because when we're dealing with donors, we have these tiny little segments of whole blood instead of actually having a whole tube.
Okay.
And they've been typed already, so when we're retyping the donors, so we're just verifying the tag on the bag is truly what it is.
Okay.
So that's forward typing. We're looking for what antigens are on the cells using known antibodies. Then the second part is reverse typing. We are finding out what antibodies are in the plasma using known antigens. So I've got A1 cells and B cells. So I know what's in this tube. I'm going to take this tube and put it with the plasma. I'm a type A. So if I put my plasma, which has anti-B, in a tube with A cells, nothing's going to happen. If I put my plasma that has anti-B into a tube with B cells, I should get agglutination. Reverse typing is always only done in test tube. So, forward and reverse typing. Forward, A and B, we also use AB. And for us, it's kind of a check for you as you're learning. A lot of places have gotten rid of it. Some places still use it. Follow your facility's procedure with what they require. We use it because if these two turn up negative, but your AB turns positive, you missed something. You know, you may have forgotten to put reagent in or something went wrong. So we still do it for you guys. So forward reaction, cells tested with anti-A and anti-B. Reverse reaction, serum with A cells and B cells. And it's always A1 cells. We'll talk about A2 cells at a later point. So it's A1 cells and B cells. So I mix up. I got anti-A with these, with the patient's cells, anti-B with patient cells, and they're both negative. That looks like an O. But we do the reverse to verify, reverse typing. Patient's serum with A1 cells is positive, serum with B cells is positive. So this patient had anti-A and anti-B in their plasma, which makes sense to be an O. So when you see these reactions, don't just memorize what they should be. Think through. In this tube, the anti-A reacted with the patient's cells, so I know that patient had A antigen. It didn't react with B, so that patient did not have B antigen, so therefore that's an A. Going backwards, patient's serum against A cells did not react. Patient's serums with B cells did react, that's an A. So when this is an A and that's an A, I can call this an A. If something happens and they're not the same, they don't make sense, where forward typing and backward typing don't match. Remember last week I said they don't make sense? Sometimes they just don't make sense, in which case you have to stop and figure out why they don't make sense. That's what would happen at this point. You can't say, well, it's probably an A, that would make most sense. First thing we're gonna do is go back and redo it. Chances are really good, you just mixed up your reagents and your tubes, which is why we do that in class versus in the lab. We want to make any of those mistakes you're going to make, we want to make it in our lab here and not your lab out there. So results of forward typing and reverse typing must agree. If forward and reverse typing do not agree, you must find out why. If you cannot figure out and find out the why, if you cannot resolve the discrepancy and make them match, you have to report the blood type as unknown and then transfuse group O blood until you can get that figured out. Because we can't take the risk. So here's just a funny, he's not her type, he's AB negative. So ABO compatibility, there's two basic rules you have to know. If you're transfusing red cells, you're transfusing antigens. And if you're transfusing plasma, you're transfusing the antibodies. So if you keep those two things straight, if I'm giving red cells, I'm giving antigens. What is safe to give to that person? What are those person's antibodies and that's safe? And then vice versa for plasma. So if I'm going to transfuse somebody A red cells, I can give A antigens to anyone with no anti-A. Okay. So therefore, I can give them to group A or group AB. If they have any A antigen, they should not have any A antibody, and I should be able to give A cells to them. B cells to anybody with no anti-B. B and AB. To transfuse AB cells, I'm going to be giving both A and B antigens. So they can't have any A antibodies or B antibodies. So pretty much AB cells can only go to group AB. Okay. However, AB is the universal cell recipient because they already have both antigens. So they're not going to ever develop. They can get A, AB, B, or O. They can get any of the types of red cells. If I'm going to give O cells, I have to give, O cells have no A, B, or antigens. I can give them to anybody. They can be given to O's, A's, ABs, or Bs. Group O is the universal red cell donor. However, as a recipient, Group O can only receive type O blood because they have anti-A and anti-B in their system. So they can only get type O blood with no A or B antigens. When it comes to plasma compatibility, O plasma can only go to group O because they have anti-A and anti-B in their plasma. So they can only go to people with no A or B antigen. Whoops, I went backwards, sorry. However, group O, because they don't have any antigen, they can receive anybody's plasma. So if you're going to transfuse A plasma, you're transfusing anti-B antibodies. So you can only give them to people without B antigen. So A and O. If I'm giving B plasma, I'm giving A antibodies, anti-A antibodies. I can only transfuse them to anybody that does not have any A antigens. So that's B and O. AB plasma, because they have both A and B antigen, they have no antibodies in their plasma. Their plasma can go to anybody. AB, it's their universal plasma donor. So AB people are super, super precious because of their plasma when we have donations. However, group AB can receive only AB plasma because A plasma has anti-B, and that's going to attack their cells. B plasma has anti-A, and that's going to attack their cells. And O plasma has A and B that will attack their cells. So they can only receive AB plasma. So this is just kind of fun and goofy. There's all these studies out there. Oh, your personality, and you know what's your. I think the next one talks about 4 blood types for 4 different diets. What your diet should be to be healthy. It's kind of crazy. During COVID, beginning of COVID, you guys all heard that. Oh, this type gets it worse. Like we're seeing more people sick with O. Well, look how many more people have O, right? I mean, O is the largest of the blood types. So yeah, you should see more O people than, you know. So people are always quick to jump on that. I belong to a couple groups on Facebook, different health groups. Like, I wonder if there's any link. Who else here is a type O? Well, I'm an O, I'm an O, I'm an O. I'm like, guys, O is the most common. Followed by A. Yeah, we're not going to see as many B's because there aren't as many B's in the pool. So anyway, this stuff is fun. There are books out there. It's goofy. I don't really buy any of it. So like this is just different. You can go look through like the different, what you're supposed to eat for health. ABO inheritance. So now let's get back to our genetics a little bit and talk about inheritance. A and B genes are on chromosome 9. They're codominant. O is actually the lack of. If you look back at this picture, you can see that what these are, they're sugars. O doesn't have any of the sugars, any of the terminal sugars that makes the A and the B. So ABO phenotypes, we talk about phenotypes, it's what we see, right? Phenotype A, A1. I'll get into A1s and 2s later. A1 can either be an A1A1, an A1A2, an A1O. A2 could be an A2A2 or an A2O. B could be a BB or a BO genotype. A1B is an A1B, A2B is an A2B, and O is always an OO. So the phenotype versus the genotype, homozygous versus heterozygous. We're kind of getting back to that again, right, when we're doing this. So possibilities of an AO mating with a BO. So you can give the A and the B for an AB. You can give the A and the O for an AO. You can give the B and the O for a BO, or the O and the O for an OO. So you can have an A and a B mom have 4 different blood types of kids. It's pretty, I mean, it's just kind of crazy, but that's just how it works, right? And we don't know. A lot of times the moms are A's and B's. So I might ask you, I will ask you actually, I have an A mom and an AB dad. What are the different phenotypes and genotypes? And I expect you to draw these out with what they are at that time. We'll talk about that in a few minutes. So possibilities of an AB with an OO. So again, you know you're an AB and you know you're an OO. So that's really good. There's only one Punnett square. A and the O is an AO, A and the O is an AO, B and the O is a BO, and B and the O is a BO. So it's 50-50, A and B. Now there's a CIS-AB phenomenon. I will never trick you. I never expect to see this in a Punnett square, but no, it does kind of exist. I've never seen it in real life. I don't know where they found it. But what happens is during meiosis, the A and the B genes end up on the same chromosome during the crossover. And so what you have is you have an AB and a dash. So one chromosome has an AB and the other one has nothing. Super, super rare. They're going to look like an AB. But this is what it looks like. So for example, if they like most of the time we think AB people have to give the A or the B, but you can end up with this O blood type because the A and the B go together. Okay, I will never trick you on this, just to know it kind of exists, it's out there. If I ask questions about mating and what kind of offspring, I think I've been put on the homework because I have people try to put this on their homework. Like, stop, no. I will never try tricking you with this. I will never try tricking you with who's the daddy. I will flat out say there is one question on your homework that talks about the daddy, right? But I will never trick you. I will always tell you if I'm expecting something different. Yeah.
I just want to say that note is not on the assignment.
It is.
Is it? I didn't.
It's on.
The next worksheet.
Is that the next worksheet? Which homework are you talking about?
Oh, I was doing assignment 2, question 2.
Yeah.
Where dad is AB, and I wrote that as one of the options.
Oh, okay. Yeah, you never have to put that. Assume I'm always straightforward with you with all that and not going to CIS-AB or who's the daddy unless I tell you. The one homework this next week, talks about you have the mom, the dad, and 3 offspring, and one of the offspring just does not make sense. So then you have to tell me like, so it's, I think, I used to say mailman, but nobody knew, or the milkman or something, and nobody knew what I was talking about, because that was like really old. The old joke was, you know, well, Prince Harry looks like that other guy. Remember, there's all this like theory out there that he belongs to the other guy. I can't think of his name right now. But it's milkman or donor or, you know, something else is going on. That's what I'm looking at. But most of the time, I'm not looking for this to say, okay? Just kind of know it exists out there. So some characteristics of ABO antibodies, also known as isoagglutinins, naturally occurring, sort of. What happens is when babies are born, they don't have any of these antibodies, and these form due to just the normal environmental bacteria, dust, things like that that they're exposed to. So you develop the antibodies against these things that are the ones you don't have. Titer runs 128 to 256. Doesn't really mean much for us. They're absent at birth. The antibodies are absent when you're born. Predominantly are going to be IgM. So predominantly they're going to be cold reacting, large IgM. They do have some IgG components, but we're not going to worry about those just yet. The IgG components usually occur after an exposure of some sort. We sometimes see that mother to baby. Like for example, if mom is a B and dad's an A, and let's say mom's a BO and dad's an AA, and so the babies keep getting A and mom has B, mom can produce some anti-B IgG after the second baby, after the first baby, after the exposure. So we'll talk about that when we get farther along to HDN, but for the most part, IgM, mostly IgM. They have direct agglutination because they're so big, they can grab multiple cells and agglutinate in saline without having to do anything special. That's direct agglutination. Optimum temperature, less than 30, but they do react at any temperature. So that's something to remember. They will cause problems in the body, even though their optimum is less than 30. Very good complement activators because they're so big. Characteristics of ABO antigens, they're oligosaccharides. Certain words, I should not have to say certain words, that's one of them. Lymphadenopathy, lymphadenopathy. I have to say it like 3 times if I say it right. That's another word I should never have to say. So they're pretty much the sugars. This is sugar attached directly to the lipids on the red cell membrane. So they actually attach to these lipids and they stick out from the red cell membrane. So they're on the red cell membrane. There's over 1 million ABO antigen sites per red cell. It's a lot. So you can kind of see how tiny they are. Because you know how small a red cell is. Now there's over 100 of these. They're soluble. That means if you have the right makeup, they are present in saliva, plasma, and other secretions. That optional lab we're doing, is that next week? Whenever it comes up, the lectin and saliva lab, we'll find out if you are a secretor and if you carry this in your saliva. About 80% of people are secretors, and so it will be in their saliva. But these antigens are capable of being in the saliva. They're soluble. They're often expressed on tissues other than RBC, so that's why it's really important when you're thinking about organ transplants, which ones have the antigens and which ones don't, and which ones are going to react and have problems. At birth, they're only moderately well developed, and we'll get into why here. So, ABOs are built on the precursor chain. So this is the cell, the sugar's on the end. This is your precursor chain. The F gene, or the H gene, adds this fucose to the end. So this is the precursor. This is the fucose that's added because the H, you can see A, B, and O all have that fucose. It's an L-fucose, it's fucose, and so it provides a fucose to this terminal sugar. Then the A gene, if you have the A gene, you're going to end up adding this N-acetylgalactosamine. You need to know this. You need to be able to recognize Na for the acetylgalactosamine is for your A gene. That's this sugar here. The B gene adds D-galactose to the terminal end, and that's your B gene. That's the differences between the different genes, or the different antigens on the red cells. Depending on if you have, everybody should have the H gene. There's a small population we'll talk about in a few slides that don't. and then you either have the A gene or the B gene or just the H gene, and it'll determine the terminal sugars. H substance must be formed initially before the A or B gene products can be added. So you have to have this on there before these can be added. A gene is more efficient at converting H than the B gene is. The A gene is more efficient than the B gene, so that means more of the H. So a lot of people that are type A, most of them, the A is going to convert, and you're going to have most of your antibodies on your cells, or antigens on your cells are going to be A. However, B isn't as good. So a lot of people with the B gene, only about half of them convert, so there's a lot of the O hanging out. Group AB has some H substance that's converted to A and some that's converted to B, all in the same cell. Group O has all unconverted H. So, the reason we bring up the H is there's this rare group called the Bombay blood group. Very, very rare. It was found in India, and they found it in a family. And what happened is they don't make the H gene. They don't have the H gene. So because they don't have the H gene, even if they have the A or the B gene, it's not going to show up on their cells because you have to have that L-glucose in order for the N-acetylglycine or the D-galactose to show up on the cells. So, because they don't have H, A, or B, when we type them, they're going to look like O's. But they're going to have anti-A, anti-B, and anti-H in their plasma because they don't have the H gene. When you lack all these things, the only cells that will be compatible with Bombay are from another Bombay. Because, let's go back to that picture, if they don't have this gene, if I give them this blood, it's now different from them. And they're going to attack it because they don't have, they have anti-H, anti-A, and anti-B. On the next one, right? Yes. They're only, red cells are only compatible with other Bombays. So Bombays go into the rare donor file, we have some frozen. Frozen isn't always the best, but we have some frozen at the Red Cross if somebody Bombay comes in. The problem is when they come in, we're going to type them and they're going to look like an O, unless we know. Because they're not going to type with anti-A, they're not going to type with anti-B, so therefore they're going to look like an O. Best source for compatible red cells are going to be siblings in this situation. Yeah?
How would you figure out the Bombay?
You don't. Not until they're having a reaction to the blood you're giving.
Oh, that sounds fun.
Mm-hmm. Yeah. Thankfully, it's so rare and it's familial groups, people know it's in their families.
Mm.
So it could be problematic if somebody is adopted and we don't know their family history. So, Bombays, this is what it's going to look like. Anti-A negative, B negative, A cells 4 plus, B cells 4 plus, going to look like an O or Bombay. Could be either one. That's how they both look exactly the same. So what we do is we add this lectin H. So a lectin is a plant-based solution that works like an antibody. And what we do is we have this anti-H, it's lectin H, but think of it as anti-H to the tube, and you can see here, type O's, it's going to react. Bombays, it's not going to react because they don't have that terminal glucose sugar to make it an H. We do not do this routinely. If we start having a reaction, this might be something the reference lab does, like, what's going on? Why are they reacting to the old blood? What's happening? That might be why. So, lectins, like I said, they're plant extracts that have antibody-like properties. Lectin H is this Ulex europus, and it's going to include a H. Lectin anti-A1 is Dilocus biflorus. So, you have to recognize, if you see this somewhere, it's a good board exam, good exam question. Lectin A, think flour. So when you're looking for an anti-A1, lectin A1, think flour. It's going to include your A1 antigens. Some labs are going to use this. We're going to talk why in a minute. So, subgroups. Leads us right into subgroups. Normal expression of A antigen is A1. So you can see There's hardly any H's. So all the antigens that were H's on this red cell converted to an A. A is really, really good at converting those red cells. A1 is. Now, there's a subgroup A2. About 20% of us are A2 subgroups. And what that is, we only are going to have about 10% of our H converted to A. So we still have a lot of H floating around in there. Clinically, not significant. Can cause problems sometimes with laboratory testing at room temperature. So subgroups are caused by decreases amounts of antigens on the red cells. There's a gene that is going to, so what we'll see is these decreased amounts, genetically transmitted, more commonly seen as subgroups of A. That's going to be the most common one we see, the subgroups of A. Cells do not agglutinate with lectin A1. They're going to agglutinate with your anti-A, the blue anti-A, but they will not agglutinate with your lectin A1. Because that's only good just for the A1 population, which honestly is 80% of the population, so that is good most of the time. Some subgroups produce some anti-A1. Not a big deal because it's cold reacting, but it might show up as a positive in the test tube, and we'll have some examples coming up. So subgroups in transplantation, ABO antigens, like I said, are on organs as well as red cells. So people who are subgroups have a lot less antigen on their organs, so their organs are better for transplanting. So it's kind of a bonus to having a subgroup. I am a A2. I have a lot less A antigen on my organs, so there are chances that I could actually give organs to B's and O's. A little bit of a bonus. However, the recipient usually has to undergo repeated plasma exchanges to lower that titer before, because we have that titer hanging out in our body. So we do plasma exchanges beforehand if we need to. So this is what we're looking at. We're looking at people who are subgroups, forward type is an A, but their serum agglutinates backwards. So this is where it's really important. Look at the forward, look at the back, make sure they both match. In this situation, if forward's anti-A with the cells is 4+, anti-B is negative. Looks like an A. But then when I do my back typing, serum plus A1 cells is a 2+, but that should be a negative if we think it's an A. And then that one works, but okay, now we can't interpret that until we figure out what's going on. So in this situation, we're going to test the patient cells with this lectin A1, and you can see we test it against an A1 for control, nice 4+. We test it against the patient, it's a 0. Let us know. We got a subgroup going on here. So the most common in a subgroup is going to be A2. So 80% are A1, about 20% are A2, and then there's like less than 1% with the other subgroups that we'll talk about. So what we do is remember I said we have A1 cells we use most of the time? We also have A2 cells in the lab. So if we do have this reaction where the patient's serum is reacting with our A1 cells, we'll grab the A2 cells out to see if they react. One thing to note when you're looking at this, we're trying to figure out, like, when you're writing these down, this isn't going to be written as big and red for you, right? You're going to have to figure it out on your own. When you're looking at this on your own, it's always going to be the one, it's almost always in the plasma that's causing the problems, and it's always going to be the lowest number, whatever's the smallest, because we expect these to be forwards down here. So those are the ones always to question first. What might be off? The one that doesn't make sense. So this is what it looked like before. Anti-A was 4, B was, so it looks like an A forward, backwards. If I have serum reacting A and 1 cells, it looks like an O, but this one's smaller. So instead of A1 cells, I'm going to swap these out for A cells, A2 cells, and now I've got A forward, A backwards. Now I can call it an A. I fixed my discrepancy. I was able to prove it with those A2 cells. Other subgroups that exist, A3, that's going to be a mixed field, AINT, AMIM, AEL. The only other one we really worry about here is this AX. Okay? AX types as an O, but will agglutinate with anti-A, B. So we have to remember that primarily with donors. If I gave an AX subgroup to an A donor, it can make them really, really sick. It can also cause misleading blood types. It's going to look like, I'm sorry, if I give it to an O donor, so it's going to look like an O. If I gave this to an O donor, it's gonna cause problems. So what we do, AX has very few A antigens on it. It's not good at converting, so it's very, very few. The red cells type is an O, but it has but there is a strong anti-A1 present in serum. Serum types is an O. Could be problems with typing, it looks like an O. So what we do is it's really important on donors. This is primarily for donor units. Whenever we have the type O, we're always going to use A comma B instead of the A and the B. We'll talk about this when we get to lab this week. I got pictures on the boards upstairs. So we're going to do, if you do A and a B, it's going to look negative. But then if I do A comma B, it's positive, and I know it's an AX. So this is how we verify our donor bags. Donor system, centers have to confirm all O's with A comma B when they get to our lab. Every time we get a bag in, we confirm every single donor bag. We have to use AB also, it's part of the FDA regulations. Not necessary for us to do our donors, our recipients. If they look like an O, we're treating them like an O. It's not gonna hurt, they're gonna get O blood. O blood's not gonna hurt, right? So it's only the donors that we're really picky about with this. So in conclusion, ABO, most important blood group system. ABO compatibility between donor and recipient is crucial. Subgroups may present typing problems. When typing, discrepancies must be resolved, and if you cannot resolve the discrepancy, must transfuse O red cells and